All right, good afternoon. So when we ended the last time, okay, we finished up metabolism and we started chapter 10, genetics, biochemistry of the genome, or I can't remember exactly what your book title's that. We talked about the structure of DNA. I want you to take a minute, take some scrap paper, and see how you do with this. Put in as much detail as you know, all right? And this is a good study technique. You see a figure that I've shown you in class, see if you can replicate it, see if you can write it out yourself. That's how you test to see if you really know something. I don't have all the detail memorized. Yeah, see if you can get the basics in. Use some scrap paper. Yep.
It took a few tries to get the.
It does.
Is it? I have to like read it. Wait, that's not right.
Add the directionality.
Like which one?
Yeah. Yeah.
My sister's in art school, so she got all the art stuff, you know. No, he's
Some of you are peeking. Don't look back at your slides. Gotcha. All right, see if you can do it without looking.
University project. Most likely. Probably. Yeah, it's more. Submittal. Younger sister.
Okay, my hope is that you can get about like to this level. Maybe you have more detail than that. Maybe you had less. But first of all, knowing what does that mean? A nucleotide that refers to deoxyribose, the sugar. It's a pentose sugar. Means 5 carbons. The base. So this is a base over here, and I'm kind of weak on sketching that out. I made a pyrimidine, so it has kind of 2 rings there. You could have had, you know, whatever. If you knew, I'm not worried that you know the structure of the base, but that's where the base attaches there to that 1' carbon. Here's the 2'. Here's the 3' carbon. So you want to know label the directionality of it. This is where the hydroxyl group is. We're going to see that today. That's really important. That's where all the new nucleotides get polymerized there to that 3' hydroxyl group. And then there's a carbon here that's not part of the ring, and that's where the phosphate group is attached there. Okay. We talked a little bit about bonding. So the bonds here that are going to, I don't know, whether there's 2 bonds or 3 bonds, that's going to be the other base on the complementary, the other strand of DNA, right? These are hydrogen bonds. I drew 2 hydrogen bonds there. And these other bonds over here, these are kind of covalent bond, the phosphodiester bonds they're much stronger because they're sharing electrons. Okay, you don't want those backbones to break, but every once in a while, as we'll see, those 2 strands do need to unzip or break apart. All right, so okay, good. And you could have drawn it, right? The strand here on the other side is going to be anti-parallel, right? So there, you know, you have to kind of draw it facing the other direction, which is kind of a good exercise too. So, okay, any questions from last time? We had talked about structure of DNA. We started talking about RNA, and we're talking about the differences between RNA and DNA. We've got a ribose sugar instead of deoxyribose, really similar, but just one place there. And of course, RNA is going to have the base uracil instead of thymine. Other differences, where DNA, of course, is double-stranded, almost always, except in some viruses. RNA is almost always going to be single-stranded. Now, there are some exceptions to that. Sometimes, as we'll see, RNA can kind of fold back on itself and maybe bond to itself, at least transiently, sometimes for longer than that. So we do see that sometimes. And of course, there is, in some viruses, are double-stranded RNA. Okay, so we don't typically find, we don't really, even if RNA kind of folds back on itself like this, we typically don't consider it double-stranded, but it can be transiently so. But it can bond to form a 3-dimensional structure. Okay, and then recognize there are different types of RNA in a cell. RNA primers, we're going to talk about their importance. They are important in when DNA copies itself, a process known as replication. These are going to be temporary, and they're really short. They're like 10 to 20 base pairs. Okay, so just really short segments. So in terms of the total RNA within a cell, this is a really small, almost negligible amount if you're looking at all the RNA in a cell. Messenger RNA, that's sort of the most famous kind of RNA, the kind that we think about because for following the flow of information, the messenger RNA is really important. Its job is to carry the DNA message. We'll say carry the message from DNA to the ribosomes so that the correct sequence of amino acids are put together at the ribosomes. But actually messenger RNA is pretty transient. It doesn't last very long until it degrades. So it's only about 4% of the total RNA in the cell. And I should mention how does a cell make RNA? It's made by that process known as transcription. We'll get to that process today. No matter what kind of RNA is being made, it's always going to be transcribed from one of the strands of DNA. Transfer RNA, that's about 10% of the total RNA in the cell, which is kind of surprising because these are pretty short pieces of RNA, maybe like 75 base pairs long. But their job is to deliver the correct amino acid to the ribosome, and that's going to happen during translation. So we often draw tRNA like this if we had it like kind of flattened out, but we really think that it's all kind of, if you look at all the bonding and the three-dimensional structure, that it would kind of look more like this. So in our old textbook, they always used to use this kind of bent shape icon to signify the tRNA. And of course, the amino acid is going to be attached at one end. And down here, these three bases, we call this the anticodon. Okay, so we'll talk more about that later, but that's going to match up with triplets of bases on the messenger RNA. They're going to be complementary to the codons in the messenger RNA. Okay, so what kind of RNA is left? What's the other kind of RNA? Right, ribosomal RNA. It's the one that, I don't know, everybody forgets about. I do sometimes, but that's about 85% of the RNA in the cell. Okay, and this is the dominant molecule in the ribosomes. Okay, so we've talked about ribosomal structure already. Of course, this is where protein synthesis is going to be taking place. What's special about ribosomal RNA is that it can be catalytic. Okay, so it can act like an enzyme. And when it acts like an enzyme, what do we call it when it acts like an enzyme? Anybody familiar with that? Yeah, right, good. We call it a ribozyme. Okay, and so ribozymes are catalyzing a lot of aspects of protein synthesis, like peptide bond formation and the cleaving of peptide bonds as well. Okay, okay, good. So again, all the RNA is going to be made by transcription, and we'll talk more about that probably later today. Oh, I didn't post this, but this is kind of just to appreciate the complexity of ribosomal RNA. This is the 16S piece of ribosomal RNA from a particular bacteria, quite complicated. If you look at the 3D shape here, the ribosomal RNA is brown, and then that purple that's really closely associated with it, those are different proteins that we find that make up the ribosome. So again, just to appreciate the complexity of that molecule. Okay, we've talked a little bit about structure of genomes already. So a little bit of review here, and we're going to compare how that some differences between prokaryotic chromosomes and eukaryotic chromosomes. Usually, so remember, prokaryotes are the archaea and the bacteria. Usually, the archaea and the bacteria are usually only going to have one. Occasionally, or two, usually one or two. Where in eukaryotes, they're going to have many. Okay, so for us, we have 46 chromosomes. Generally, the more chromosomes, the more complex the creature, but there's a lot of exceptions to that. Okay. Prokaryotic chromosomes are going to be circular, and they can be, not just, they can be these big, long, kind of complicated structures, but they're circular, okay? And that's going to be a more stable way for them to store their DNA because the ends aren't exposed. Okay, our chromosomes are linear, and we have to have ways so the ends don't get kind of digested by nucleases in the cell. Prokaryotic chromosomes are also haploid. That means there's one copy of each gene. And we know eukaryotes like us, we are diploid. Okay, we have 46 chromosomes, we have 23 pairs. Okay, so we have 2 copies of each gene or of each chromosome. There is differences in how the DNA in the chromosomes is stored. I'm not going to get too much into that, but there's just way more DNA in eukaryotes. They have ways of winding it up around histone proteins when it's not being used. We see some similar things in prokaryotes, but it's not quite the same. So I don't want to really go into that too much. One last thing, where the chromosome is located. Okay, we talked about this before too, in the nucleoid region of the cytoplasm. Okay, and of course for eukaryotes, that's going to be in the membrane-bound nucleus. So again, this is just a region in the cytoplasm where the chromosome, maybe in some plasmids associated, tend to be. It's not surrounded by any kind of membrane. In addition to the chromosome, prokaryotes especially, or I should say prokaryotes, are more famous for having these smaller circular kind of molecules of DNA called plasmids. And I was looking this up a little bit before lecture, and it turns out that there are plasmids in eukaryotes as well. Plants and fungi especially, but they have something that's kind of comparable to plasmids, but we kind of think about this as being more of a prokaryotic thing. They replicate independently of the chromosome, and typically what kind of genes do we see on plasmids rather than chromosomes are genes that are absolutely essential for the cell to survive. So we're not going to see enzymes for glycolysis on a plasmid. Those are going to be on the chromosome. But we may see other things or different kinds of plasmids that we see. For instance, fertility plasmids. We're going to talk about those more later when we talk specifically about conjugation. I've mentioned that a few times before, but they carry the instructions for conjugation and the making of a pili. Okay, a pilus is essential for a cell to share a plasmid by this process called conjugation that, again, I mentioned, but we'll talk about at the end of this unit. Resistance plasmids. May carry some gene that will enable the bacteria to survive in the presence of some antibiotic. And then there are others, but one other one I want to mention are virulence plasmids. We're going to talk much more about virulence when we look at the process of infection, maybe a few weeks from now. But these are genes and proteins that would allow an organism to establish itself in a host. So it might be the ability to outwit our phagocytic cells. It might be the ability to make some kind of toxin that makes it successful to infect a host. Okay, so we'll talk more about those. Okay, so that's kind of the basics. That was kind of chapter 10, the basics, as we lead up to thinking about these processes and how DNA is made, how RNA, and how proteins are made. Okay, so looking ahead at chapter 11, the functions of genetic material. There's a little bit, we want to, kind of an overview that we want to look at, as well as there's another, we talked about a couple experiments last time, another really famous experiment that I want to walk through a little bit. And then we'll get into sort of the nitty gritty of DNA copying itself. We call that replication. There's some enzymes that you're going to want to understand their role. Transcription gets a little bit more complicated because there's kind of a start and a stop to it, as well as translation, protein synthesis. How does the process start? How does the process stop? We'll see how far we get today. I probably won't go beyond protein synthesis, and I'm not sure if we'll get that far. But then looking next Monday, we'll likely get into talking about a few operons, gene regulation, why that's important, and then ways that DNA can be changed within a cell by either mutation or some kind of horizontal gene transfer. So right now, I'm thinking that we will finish this material up by Monday, most likely, and then next Wednesday's lecture will not be included on the following Monday's exam. So the way I have it set up of chapter 11 homework being due on our exam day, I think that I'm thinking that will work out about right. We will start viruses next week, but that won't be on this exam too. That's on the horizon. Okay. All right, so hopefully this is review. I know some of you have told me, I don't remember anything I did in biology. Okay, well, if it's not review, I might need to spend a little bit more time on it. But the central dogma of genetics, the flow of information, the sequence of bases in DNA, we call that the genotype of an organism, okay? Sequence of bases. And then, of course, when that is replicated, when the DNA is copied, the only time that happens is when the organism is, when the bacteria cell is getting ready to divide. Okay, it copies its DNA, and then each copy, each daughter cell has a set of DNA, obviously. Okay, so the cell doesn't need to replicate its DNA every time before it transcribes the DNA. Sometimes students have that misconception. So, but that sequence of bases in the DNA, by a process known as transcription, we get a messenger RNA template, and then that is what's read at the ribosomes to determine the sequence of amino acids that make up a protein. And then what proteins an organism can make, that determines the phenotype of an organism. So for us, that's things like eye color or, a million other things, skin color, whatever. For a bacteria cell, that might be, phenotype may be, can they metabolize lactose or not? So, whether they can make the enzymes to metabolize lactose, that's kind of an example of a phenotype for a bacteria. Now, that is not set in stone. We recognize now that sometimes there are different environmental influences that will affect whether a cell maybe will show one phenotype or another. Certainly as true in humans, it can be, for like height, like how high we get, that can depend on nutrition at a young age, for instance, some environmental condition or whether you were exposed to a lot of cigarette smoke, or whether you smoked yourself. That can make a difference whether you develop lung cancer or not. So certainly there is a role in the environment to determine the phenotype. Okay, so we talked about last time Frederick Griffin's very famous experiments that showed how there is some genetic material that can be transferred from one cell to another. And then Avery, McLeod, and McCarty showed that that genetic molecule was actually DNA and not some protein. And then, of course, Watson and Crick and others contributed as well, like Rosalind Franklin, into elucidating the structure of DNA, but they didn't really do any experiments as to how DNA was replicated. And they had kind of an inkling, kind of a guess as to how that happened, but we didn't really have a definitive answer how that happened. So after that 1953 paper of Watson and Crick, there was 3 theories as to how DNA was replicated. And those 3 theories were the conservative method, the semi-conservative method, or the dispersive method. Okay, so with the conservative method, that was the theory that the original DNA was conserved. So if you have, if the light blue is the parent DNA, and then the 2 cells that result of that, for instance, would one cell be the same as the parent, be conserved, and the other cell be totally new? Was that correct, or was there a semi-conservative? So one strand of the parent DNA was conserved while one strand was new? Or was it by this dispersive method, where basically, it was either one strand or the other, replication happened in a very piecemeal way, and the offspring are a mix of the old DNA and new DNA that was synthesized. So we didn't really know. You can see here with the 3 different methods, how maybe the first generation of cells would look and the second generation. Well, in 1958, an experiment was published by Messelson and Stahl. Again, this was 1958. And I want to go through this because this has been called the most beautiful experiment in biology. And the reason it's known as that is because not only did it prove which method of DNA replication was correct, but it also disproved the other 2 theories. Okay, so it was very quickly accepted, unlike other theories that might be 40 or 50 years till we get the evidence that they're correct. This really convinced people right away. And this link down here at the bottom was a YouTube video that I stumbled across a few months ago, interviewing Messelson and Stahl. This was just a few years ago. And it's just kind of the cutest video, these old men kind of thinking back to how they did this experiment and what their life was like, and how different science was back at that time. It's just kind of amusing if you're interested in that sort of thing. So that's just for the fun of it there. So if we look at this experiment, what they did, they recognized that DNA is very rich in nitrogen. You have these bases, these nitrogenous rich bases. So they grew up some bacteria cells in a heavy isotope of nitrogen, nitrogen 15. Okay, so it has an extra neutron. It's not radioactive, it's just a little bit heavier. And then they would extract that DNA from organisms and spin it down in a centrifuge, and it would make a band. It would kind of sink pretty low in the centrifuge, that DNA that had that heavy nitrogen in it. And so that would, whichever hypothesis was correct, okay, that's the way the DNA would grow to begin with. So then they took some cells that were growing in heavy nitrogen and they switched them to a medium that had regular nitrogen in it, or we'll call it light nitrogen. And then after one generation, so they had to know the generation time of whatever bacteria they used, I don't know if it was E. coli or what, and they extracted the DNA, and they found that it had kind of a hybrid band. So the DNA wasn't quite as heavy. So right away, they could tell that this conservative method was not correct, because if it was conservative, they would still have a heavy band, and then the new DNA would have the light band. There wouldn't be a hybrid band. So they could tell after one generation that the conservative method was not correct. But with both semi-conservative and dispersive, they would both have hybrid bands. So they had to go out a second generation and do the same thing. And here they found two bands. So they had the hybrid band, and then new on top of that would have a lighter band, and that matched. That would be what we would expect if it was semi-conservative. And to make sense of that, you can go back to here and you would see, oh yeah, here would be a light band, heavy band and a heavy band. This would be hybrid, this would be hybrid, and these would both be light. Okay, so here, we're still gonna all have hybrid. All right, so what they found anyway, so the results matched what you would expect for semi-conservative replication. Okay, so here's a link to a YouTube video that probably explains that better than I just did. So that's just a couple minutes. Again, it's not easy for me to show these in class because the sound doesn't come out, but again, if you're interested or maybe need to hear that again, that's a good link. So getting into kind of the minutia, like we know, okay, semi-conservative replication, what does that mean on a more molecular level? So DNA is a polymer, right? It's made up of lots of monomers. And it's actually not constructed of nucleotides exactly. Those new monomers that are added to this growing DNA strand are actually triphosphate nucleotides. Okay, so bear with me, the terminology gets a little bit cumbersome here, but these nucleotides actually carry with them the energy that's necessary for the polymerization reaction. Okay, so we have these triphosphate nucleotides, floating around this nucleoid region of a bacterial cell, and they have, 3 phosphates, high energy bonds between each of those phosphates. Okay, so here, for example, if the base was guanine, we would call this a guanosine triphosphate deoxyribonucleotide. Okay, so here's the shorthand for that. And like I said, it's a little bit cumbersome, but just recognize it's carrying actually 3 phosphates. And then remember, okay, so we have the base, we have the sugar, the nucleotide, that's the base, the sugar, or the phosphate. And if we call something a nucleoside, that's just the sugar and the base. Okay, so we're gonna talk about some nucleoside analogs later on. These are drugs that, again, they kind of are similar to the deoxyribose and then one of the bases, but aren't exactly the same. Okay, so nucleotide, nucleoside, triphosphates, deoxyribonucleotide. Okay, so these triphosphate nucleotides are going to be, again, if you look at that polymerization reaction, here's one strand of DNA, and of course you'd have the other strands over here, maybe, and then this would probably be a cytosine here. Here's the 3' hydroxyl group, all the new triphosphate ribonucleotides are added on to that end. The polymerization, there's enzymes that actually cleave 2 of those phosphates off. Why not 1? Why 2? Well, when you cleave 2 phosphates off, that's extra energy that makes this a really fast reaction, polymerization reaction. And now we've got that new one added on to that DNA strand to make it longer. So a little more detail. So in bacteria, remember there's a circular chromosome, and where that replication begins is a place called the origin. Okay, the beginning. All right. It makes sense, I hope. Okay, so that's the site of initiation of replication. So you can see down here, they put there's a little box here on that circular chromosome, just like, okay, say here's the spot that the origin of replication. Usually there's going to be some kind of signal there, so the enzymes know where to attach. Usually there's going to be a sequence that's rich in adenines and thymines, because remember, there's only 2 hydrogen bonds that are connecting the adenines and thymines. So those are going to break apart a little bit easier than what the, if there was 3 hydrogen bonds there. And we'll say replication is going to proceed in 2 directions. Okay, we call that bidirectional, right? So we've got, you can see here, you've got a replication fork opening up here and a replication fork opening up here. So again, it's going to go in both directions. And this enzyme helicase, this is important. That's what's going to unzip the 2 strands of DNA in readiness for replication. Okay, so we say unzips. Sometimes you see the term melts. Okay, the 2 strands of DNA. Helicase separates, it breaks those hydrogen bonds there. And I should mention, I'll come back to this in a minute, but in eukaryotes, they're going to have multiple origins. Okay, prokaryotes, there's only 1. Eukaryotes have that DNA, that chromosome is going to be so much larger that lots of different origins are going to open up at once. Because if they only did it in one spot, it would just take too long to replicate all the DNA. Okay, so again, eukaryotes, multiple origins. Okay. That last enzyme that I want to talk about, topoisomerase 4. This is important to resolve something called a concatemer. So this is something that's unique to prokaryotic replication because it's got a circular structure. And so you'll see here in this step, once replication is completed, actually those 2 rings that are left are actually linked to each other. And it's hard to kind of wrap your head around how that would happen exactly. But again, that's when 2 circular genomes are linked. Okay, so there has to be a way to basically unhook them from each other. So that's what this enzyme does. It resolves those concatenators. It makes a cut and seals it back up again so that we have two new genomes, one for each daughter cell. All right, so again, that topoisomerase 4 is going to be unique to prokaryotes because the eukaryotes don't have the circular genome. They don't have that specific problem. There are different kinds of topoisomerases, but this one in particular is it's prokaryotes only. Okay, so some more enzymes that are involved in this process. The most famous one, if you can only remember one, the most important enzyme here in DNA replication is DNA polymerase 3. And it seems odd that if, you know, it's the most important one, why do we call it the third one? Well, they were named the order that they were discovered, so whatever. So DNA polymerase 3, this is the main enzyme that's moving down the old strands of DNA, and it's actually moving on both strands, but it has to go, it can only move in one direction. Okay, here we call this the leading strand because it's going to be moving towards the replication fork. That's not necessarily the top strand, but here it's given a 3', so we know that this is the 5' of the new DNA, and the new is always adding on to the 3' end. The other strand, which we'll refer to as the lagging strand, the DNA polymerase still is moving in the same direction, so it has to kind of move in a series that's going to go like this. Okay, so it eventually moves towards the replication fork, but in a series of fragments that we're going to call Okazaki fragments. Okay, we see that on a later slide. So again, this adds those triphosphate nucleotides and nucleotides that have the phosphates with them for the energy to polymerize the reaction. The speed here is impressive. Whoops, not 5,000. 500 to 1,000 base pairs are added per second. So astonishing speed when you think about it. And something else that this enzyme does really good, it has a really good proofreading ability. So not only does it move really fast down the strand of DNA, but it can kind of look behind it and make sure that it added the correct base pair that's complementary to the template that's there already. Okay. All right, so what are some other, and you can see here, here's the DNA helicase. Remember, that's what breaks those hydrogen bonds between the complementary base pairs, and some stabilizing proteins here that just kind of help keep the replication fork open, and so that the other enzymes can work. Okay, what other enzymes are involved? Well, that RNA polymerase 3, it turns out it can't start the new strand from nothing. Okay, we say it can't start de novo from nothing. It needs a 3' hydroxyl group to add to. So an enzyme called primase will go through and add 1' for the leading strand and a series of primers for the lagging strand. Okay, so synthesizes an RNA primer at the origin. And this is maybe, it's short, like I mentioned before, just like 10 to 20 base pairs long. Important to note, these are very temporary. Because you think about it, you can't have a DNA strand that has these little bits of RNA sprinkled throughout it. So it's only going to be there for a short while, and then some other enzymes are going to go in and replace it. And you can see here, this new strand, this is the leading strand, remember, and this is the 5' end, which is opposite from, remember, anti-parallel to the old strand. Okay, so that's the leading strand. That's pretty much it copies that leading strand until all the DNA is copied. Okay, pretty straightforward, except for have to replace the primer. On the lagging strand then, like I said, we have a series of primers. They're written here in red. So that DNA polymerase 3 will synthesize DNA in short segments called Okazaki fragments. Named after whoever discovered it. Okay, and I say short, but these are about 1,000 base pairs long. Sorry, my writing's hard to read there, but Okazaki fragments, about 1,000 base pairs. So I guess short in the scheme of things, that seems kind of long. And then some other enzymes that are involved here, a couple more. DNA polymerase 1 is going to go in and replace the primers with DNA. Okay, so you can see that right there. You can see the primer being removed, but that doesn't quite finish the job. The last enzyme important here is ligase. After the DNA polymerase gets rid of the primer, ligase is going to seal the gaps. Actually, what it does is it adds a phosphate and a phosphodiester bond. Question. I know that's hard to read. In DNA polymerase 3 synthesizes DNA in segments. Yeah, sorry about that. Okazaki fragments. I'm getting tired. My handwriting is suffering. Okay, so there's a really good YouTube animation. It's just like a minute or two long that shows this replication process. It shows the helicase working. It shows the DNA polymerase 3 working. It shows the DNA polymerase 1 getting rid of the primers and the ligase sealing the gaps. So it's just at sort of the right level. It's more complicated than this, but that's if you're a visual learner, check out that video. Like I said, it's just a couple minutes long. Okay. Couple more details that I want to mention about replication. There's another important enzyme that deals with the problem of supercoiling. And you kind of understand supercoiling if you've ever sat on a swing and spun around and around and around, like who hasn't done that? Eventually, right, the chains get so twisted up that you get bent down lower and lower because you can't pull the chains apart anymore, right? So that's essentially supercoiling. Here's another display of that. Like when you're pulling two bands apart, you get this twisting up. Eventually, the mechanical strain is too much, and you can't keep pulling those strands apart anymore. Well, this can happen in DNA replication as well. So an enzyme basically has to go in and make cuts here, unwind the DNA, and seal it back up again. Okay, so that enzyme is called DNA gyrase. Okay, that is an enzyme that's unique to prokaryotes. We have enzymes that do that too. They call them different things. They're slightly different. So again, anytime you have an enzyme that's unique to a bacteria, we have antibiotics that target DNA gyrase. Okay, we can capitalize on that. Okay, so again, that cuts both the strands, it does a lot, cuts the strands, rotates it back around and seals the strands up again. The other thing, and again, that's happening, we would say upstream here of the replication fork. One other thing that I want to point out is that there is some DNA methylation that takes place. So here's a methyl group, that's CH3. For bacteria, after replication, typically some methyl groups are added to the admins. Other organisms get methylated, but typically, I think in humans, I think it's cytosines that get methylated. Well, and we'll see how that can play a role in DNA repair a little bit later, helping enzymes to understand which is, if there is a mismatch, like the wrong base was made, that they don't actually complement each other. How does the enzyme know which base to fix? It's the strand that hasn't been methylated yet. Okay, so that methylation, and we'll look at that again in more detail later. So methylation is important there. We think it might help define the origin. So in addition to lots of maybe A's and T's, there's some methylation there. And in terms of regulation, it may make a difference as to whether that DNA gets transcribed or not, how highly it's methylated. So there's many different regulation mechanisms that happen. This just is one of them. Okay, so questions about replication. All right, let's move on to transcription then. Okay, this is the process by which RNA is made from DNA. Okay. So in DNA replication, like I said, all the DNA gets copied. But when we're making RNA from DNA, it's usually just a piece of DNA that gets made into RNA. So it's a little bit complicated. We break this up into different phases. We call it initiation. Oh, I did not spell that right. There, that's better. Initiation, elongation of the RNA strand, and then how it stops, termination. Okay, so we'll start with initiation, things that have to happen for the enzyme to start making RNA. Elongation is pretty straightforward, it just keeps making it. And then there's a couple of different ways that termination can take place, and we'll look at those. All right, so I find this graphic kind of helpful because there's some terminology that we see that we want to be able to understand. Okay, so if we look down here, this is the strand. This is a strand of DNA, right? That's being transcribed, and here's the main enzyme that's working in transcription for bacteria. Always assume that I'm talking about transcription in bacteria. When I get done talking about transcription, I'll say, oh, here's some differences in eukaryotes. But assume that I'm talking about bacteria. In bacteria, no matter what kind of RNA is being made, it's one enzyme that makes all different kinds of RNA. In eukaryotes, we got different RNA polymerases that make different kinds of RNA. But this is the main enzyme, RNA polymerase. It's moving down that DNA sequence. It's adding on, this time, triphosphate ribonucleotides with ribose instead of deoxyribose, but same idea. It carries, the phosphates with it to power the polymerization reaction. And it's adding on to the 3' hydroxyl group there, just kind of like we saw in DNA. But that RNA polymerase has to bind really tightly to a sequence on the DNA called the promoter. We'll talk next about how it does that. But first, I just want to kind of get straight some terminology with the DNA. You can see here that either one strand of DNA is going to be transcribed or the other, not both. Okay. How does the RNA polymerase know where to stop? Well, it depends where it recognizes the promoter. But say this strand, right, is being transcribed. What are some names we have for that strand of DNA? Sometimes we refer to it as the antisense strand or the negative sense strand. Why would we call that the negative sense? Because this RNA that's being made is always going to be considered plus sense RNA. If this is messenger RNA, it's plus sense messenger RNA that is able to be read at the ribosomes. Okay, so this other strand of DNA, this is called the plus sense DNA or the sense strand. Okay, you always make a positive sense from a negative, or you make a negative from a positive. It just means the positives and negatives complement each other, right? So if you take this other strand of DNA on the top here and you make some RNA, it's going to make negative sense RNA, which is not going to be read correctly at the ribosomes. Okay, so again, how do you know which is which? Well, it's whichever one the enzyme chooses, and then you name from there. Let me write in a few base pairs then to illustrate the other name that we use to describe these strands. Okay, so let's say the DNA here, let me just write in a few, is TACGG. Then the RNA that complements that would be AUGC, CC, right? Okay, and you might recognize AUG as a start codon, which again is always the first one that's read at the ribosome. So I kind of did that on purpose. But let's see. So this would be the 3' and 5'. Okay, and then if you look at the opposite strand of DNA, okay, that must be complementary to this. So, and note for the RNA, I put in U's instead of T's. The opposite strand here would be ATGCC. Okay, so the other name that we have for these DNA strands, the antisense strand, is also known as the template strand. That makes sense because when you use something as a template, that's, you know, the instructions that you use to make something else. So that may be familiar. But the other strand is, in addition to being called the plus sense or the sense strand, we also refer to that as the coding strand. And that's not as obvious, but if you look at the messenger RNA here that is made from the template strand, what do you notice? It matches exactly the other DNA strand, except for there's U's instead of T's. Okay, so the coding strand, that's the same code as the messenger RNA that's used from the other strand. So again, those terms can be a little bit confusing, but the idea is if messenger RNA is made from this other strand, it's going to be a totally different sequence as if the other strand is used. Okay, and it's not likely that both of them would actually encode for a protein. Okay. All right, so again, ask me any questions about that. I know that's kind of a lot of words. Okay, so let's look at how initiation starts of transcription. So I said that involves this promoter region of the DNA that's upstream from where transcription actually starts. Okay, so, oh, okay. First of all, before we do that, let me talk a little bit more about this enzyme. This RNA polymerase that is in charge of transcription, it does a lot, okay? If we compare it to the enzyme that's used in replication, okay, and this is transcription. Remember, RNA polymerase 3 does not separate those strands of DNA itself. It needs a helicase enzyme to do it. For RNA polymerase, no helicase necessary. Okay, it is able to unzip DNA itself. And of course, we talked about how DNA polymerase 3 needs a primer to get started from. RNA polymerase, no primer needed. Okay, of course, we've got these 4 bases for that are added during replication, and we've got uracil instead of thymine. We said that DNA polymerase 3 had a really good proofreading mechanism. It makes an error 1 out of 10 billion times. So that's pretty good. RNA polymerase is not that good. Okay, the proofreading is less efficient. We get errors here in transcription about 1 every 10,000 base pairs. But that's okay that the fidelity is not quite as good. When replication is like, it's like the blueprint, right? And if the blueprint is wrong, then every time, you know, if you copy a recipe wrong, every time you make the recipe, you know, whatever, the cookies or whatever is not going to work out. But if the blueprint is right, if the recipe is right, and you just read it wrong once, and you add, you know, baking soda instead of baking powder or whatever, well, probably the next time you read it correctly. Okay, so if every once in a while the RNA transcript is not correct, it's usually not a big deal because probably the next time it will be. Okay, so again, it's okay that the fidelity is getting a little bit worse and worse. Okay, so let's talk about that initiation. So I like this graphic. It's not from your textbook, but orient yourself here. This is representing the DNA sequence in the promoter region. Okay, remember, this is just a sequence of base pairs of the DNA, a little bit upstream of where we're going to start the transcription. And here's our main enzyme, the RNA polymerase, and it's binding perhaps to that promoter region. Okay, main thing to understand here, and I just wrote it out, the more tightly the RNA polymerase binds to that promoter sequence, the more likely it is going to then transcribe that gene into RNA. Okay, so if it binds only loosely, maybe sometimes it will transcribe it, maybe sometimes not. What depends how tightly it binds? One thing called, this is called the sigma factor. It's lowercase Greek sigma there. Sigma factor binds to the RNA polymerase during initiation and helps RNA polymerase bind more tightly. Okay, and again, this is just something in prokaryotes. Eukaryotes, we don't have sigma factors. We have lots of other things. And there can be different sigma factors that come in that will actually make the RNA polymerase bind either really tightly or maybe not quite as tightly. Okay, so that's kind of one area of regulation. Also, you'll note these two sequences, the -10 region and the -35 region. So roughly 10 base pairs up from the start site of transcription, or roughly 35 base pairs upstream. We think that those two sites are especially important for RNA polymerase binding. And in eukaryotes, we call this -10 region the Tata box. In prokaryotes, they give it a different name called the Pridnell box. Let's just call it -10 and -35, and that's fine for me. Let me show you another graphic here that looks complicated, but I'm hopefully will help you kind of pull some things together when I explain it. Okay, so this is, if you just look at this top row here, whoops. Okay, this is a sequence of the promoter regions. So these are promoter regions of operons of E. coli. There's actually like 600 different operons in E. coli, so this is just a few of them. And remember what an operon is. This is a group of genes that is under the control of the same promoter. So we're going to talk more about operons, but basically in bacteria, if there's several different proteins that are necessary to do one thing, they're kind of regulated together. Like if you make one protein, you just, you want to make the next one as well. So it makes sense that they're regulated together. So what they've done here is shown the sequence of lots of different promoter regions of lots of different operons. The lac operon, that's probably the one that you're most familiar with. Okay, and they've sequenced out this -10 and the -35 region, and they have noticed that like 69% of the time the first base is a T, and most of the time the second base is a T. So these are called consensus sequences of both the -35 and the -10 region. And what we understand now is that the more likely that -35 region, the more close it is to some consensus sequence, then the more likely that RNA polymerase is going to bind to it. Okay, so let me write that in. Genes with promoters that match consensus sequences are more likely to bind RNA polymerase. And therefore more likely to get transcribed. All right, so let's say that RNA polymerase has bound really tightly. Okay, then it's going to move down that DNA and start making RNA. Okay, we call that elongation. So here's this big orange blob, that's the RNA polymerase that's moving down one strand of DNA and making the RNA. Okay, it's adding on this 3' end. There's got to be some homework question that will ask you about directionality, and that can be a little confusing because are you talking about the old strand or the new strand? But if you remember, the new strand is always adding on to that 3' hydroxyl group for both replication and transcription, then you'll be in good shape. Okay. Again, it's talking about the template. This calls it the non-template. Remember, I also called that the coding strand. Okay. And then basically it just keeps elongating. I want to mention that in prokaryotes, well, in eukaryotes as well. Multiple RNAs can be transcribed at the same time. Okay, so as long as you've got more copies of RNA polymerase, they can just come down and keep transcribing that same gene over and over again. And think about also where transcription takes place. It's going to take place wherever the DNA is. Okay, so for prokaryotes, that's in the cytoplasm. True, the nucleoid region, but we still consider that the cytoplasm. And of course, for eukaryotes, that's going to be in the nucleus. Okay, so transcription is going to happen wherever the DNA is. The DNA has to be there. Okay, to finish today, we're going to talk about the two ways that transcription will stop. Okay, we call that termination of transcription. There's two methods, and we think that each of them occur with about equal frequency. The details are a little light here because A, we don't totally understand this process exactly, and B, it gets really, really complicated really quickly. So kind of an overview of these two different methods. One method is that involves this protein called Rho. All right, so we call it Rho dependent. Rho is a helicase enzyme. Okay, so remember what helicases do? They unzip, they separate two strands of DNA. So basically what happens, the Rho moves along the RNA, the little pinkish bracelet there, that's the Rho protein, and it moves down along that new strand of messenger RNA until we've got these kind of weak hydrogen bonds when the RNA is first transcribed, and it moves down and breaks those bonds, and basically then this is showing everything dissociating. The RNA polymerase coming off of the DNA, and the new messenger RNA disassociating from the DNA. Okay. How does the enzyme know like how fast to go and where to stop and all that? Like, I don't know. Okay, but just that this Rho protein is involved to stop transcription. And the other way does not involve the Rho protein. So we'll call it Rho independent. Another way to say that, we also call this sequence dependent, or even sometimes you hear it called self-termination. So again, we got a lot of different names for the same thing. But basically this involves when in the DNA sequence written into it, there's some inverted sequences. So another way to think about that is palindromes. So like, it's a little different, like the name Hannah is a palindrome, it's the same forward and back. When we're talking about DNA, it's a little bit different. It's kind of two sequences that will fold up and be complementary to each other. So it's kind of the same one direction, like the sequences will match up. So you see like these two sequences here, okay, they're actually a mirror image of each other, and then they will be complementary. So basically when the RNA transcribes those sequences, it can kind of fold back on itself and make this hairpin loop. And that causes some mechanical stress, we think, and causes the RNA to dissociate from the DNA. And in addition, there's usually some AU rich sequences here, which remember 2 hydrogen bonds instead of 3 that the C's and the G's would have. So this little weakness here, in addition to the mechanical strain imposed by that hairpin loop, causes dissociation of RNA. Okay, so that's termination of transcription. Okay, I want to talk about a few differences. I think maybe I will save that for the beginning of next lecture. Okay, so I do have office hours all morning Thursday, all morning Friday. If another time in the afternoon works for you, I might be able to work that out. Have a good rest of your week. Good weekend.
Okay,